So what I would like to do is I give you a little scientific presentation about a topic we have worked on, which is cell sorting in gastulation. And I will speak about that specifically because we have used, for analyzing cell sorting, we have used micropipettes. And it gives you sort of an idea what you can do with pipettes if you're interested in analyzing mechanical and adhesive features of cells during the sorting behavior. Okay, so what I will give you, what I will do in the next 45 minutes, I give you a little bit of developmental biology introduction at the beginning, and then I will show you what we have done to analyze cell properties which lead to their sorting behavior, and how we have used micropipettes in this process. Okay, so I will speak about progenitor cell segregation in zebrafish gastulation. Before I tell you what we have done on this topic, is that I quickly want to acknowledge the one person who has done most of this work. This is Jean Lyon, who's sitting down here. And all that was done in collaboration with a number of physicists. Eva Taluch is one of them. Helene Bartholomew is another one. She was a postdoc in the lab of Frank Jülicher. And all the theory I'm going to tell you about has been done in collaboration with Frank Jülicher. Okay, now progenitor cell segregation in zebrafish gastulation. Before I can tell you much about it, I need to give you a very, very little introduction into zebrafish gastulation and where cell sorting is actually happening. This is a fish embryo at the onset of gastulation and consists out of approximately 1,000 cells sitting on top of a very big yolk cell. The embryo is approximately 4 hours old at this stage. And then what gastulation is doing, it sort of transforms this seemingly unstructured cluster of cells into something which looks like a proper embryo. So you have a head structure, you have a tail structure, you have a body axis, muscle packages are developing here. This is going to be in the brain area. It has a clear anterior-posterior polarity, it has a clear dorsal-ventral polarity, and it has even a left-right polarity at this stage already, which you can't see on this picture. So there are significant rearrangement of cells leading to the formation of this structure. And we are particularly interested in understanding how that is being achieved. Now, to very shortly summarize the different type of cellular rearrangements which transform this cluster of cells into an embryo, I've put in this schematic diagram, which I've taken from Wolpert's Principles in Development, and a couple of still pictures out of a movie which Don Kane has recorded a number of years ago. Now again, this is an embryo at the onset of gastulation, and this is an embryo which has formed already a body axis pretty much at the end of gastulation. And there are a couple of cellular rearrangements which transform this cluster of cells into this elongated structure, which is the body axis. And again, so the first type of movement, so you're starting here with having 1,000 cells sitting on top of this very big yolk cell here, and what the first type of movement the embryo has to do is it has to spread the blastoderm over the yolk cell. And this movement is commonly called the Peabody movement. And it's being achieved by very specific cellular type of cellular rearrangements, and that is that cells deep within the blastoderm moving up into more superficial layers of the blastoderm, undergoing what is called radial intercalation. And by undergoing radial intercalation, what you basically do is you take the tissue and you change the thickness along one axis, and you're basically extending it along the other two axes. So it's basically sort of an intercalation movement which leads to the extension of the cell mass over the yolk cell. What I will specifically then speak about for the rest of my talk is the critical process in gastulation, and that is basically setting apart the different progenitor cells for the different germ layers. And usually you have 3 different types of germ layers in vertebrates. You have ectoderm, mesoderm, and endoderm. Ectoderm is giving rise to the nervous system, the skin, mesoderm, muscles, and endoderm, digestive tract. And what multicellular organisms usually do is they try to get the progenitor cells for mesoderm and endoderm to the inside and leaving the ectoderm on the outside. And that's a way of segregating these population of cells. In the fish, this works the following way. You have the yolk cell. Within the yolk cell, you have RNA localized in equatorial regions of the spherical yolk cell in this region. And so sort of around the equator, you localize RNA, and this RNA is coding for nodal TGF-beta signals. A signal which, once it is secreted in these equatorial regions, acts specifically and locally on these marginal blastoderm cells, and induces in these cells mesoderm and endoderm cell fates. So these marginal cells become mesoderm and endoderm, and once they're becoming mesoderm and endoderm, they're undergoing something which is commonly called an epithelia to mesenchymal transition. The cells which are not being induced, they have sort of a pseudo-epithelial structure. But the cells which are becoming mesoderm, endoderm, they're becoming mesenchymal, clearly mesenchymal, they're becoming migratory, very motile, and they're going to move in and form a second layer of cells down here, which is indicated here with a red arrow, which is called the mesendoderm germ layer. And the cells which are not going in and staying on the outside, they are becoming the endoderm. And this is really critical in gastrulation. You have to segregate these progenitor cells, and you have to order them in a specific spatial ordering where you have mesoderm on the mesendoderm on the inside and ectoderm on the outside. The gastrulation also contains two other types of cell movements which I'm not going to speak about. That is convergence of old cells towards the dorsal axis, thickening. You can see here accumulation of cells on the dorsal axis. These cells are, you have to imagine these cells are moving towards the dorsal axis around the sphere. They're meeting at the dorsal axis on both sides. They're undergoing medial lateral intercalation, and by doing that, they're extending the body axis. Very roughly what is happening during convergence and extension. But I will not speak very much about how convergent extension is being mediated in the embryo. But I will speak about how mesoderm and endoderm progenitor cells are segregating from ectoderm progenitor cells. Now, just to show you how this process looks like in low magnification, you're looking here at this little region, and you might be able to see some of these cells moving in and forming the second layer of cells on the inside, and the cells which are staying on the outside becoming ectoderm, and the cells which are going in mesoderm into endoderm. And this is the process we really would like to understand. In a slightly higher magnification, I put in this movie, which we recorded a number of years ago using a 2-photon laser scanning microscope. And what we did is we used this 2-photon laser scanning microscope to get deep into the embryo. We label all the cells here with a plasma membrane GFP and a cytoplasmic GFP. And what you can do is then you can watch how these cells at the margin, it's basically this region here, turn counterclockwise a bit. You can then watch how these mesoderm and endoderm cells segregating from the ectoderm progenitor cells and how they're forming a distinct layer of cells. What really fascinated us about this process is once these cells are coming in, and I'll show you that in a minute how that dynamically looks like, once they're going in, they never go back again. There's no physical structure separating these two cell types, but once the mesoderm and endoderm cells go in, there's no way, there's not a single cell which would ever go back into the tissue they originated from. So there's a very, very strict segregation between these cells, and this is not mediated by any physical structure or anything which is obviously separating them. Just to show you now this movie, you might be able to see some of these cells coming in here, and they're forming this layer down here, which is the mesendoderm. And there's a very clear and tight border between these two cell populations, which doesn't allow any crossing anymore, not a single cell. Okay, so we wanted to understand how that is being achieved. And the way we are addressing these problems is not starting looking in the embryo, which is very, very complex, that it's very hard to control these experiments, but reducing the complexity by establishing an in vitro assay where we can learn something about the general features, the self-organizing properties of these cells, and then going from these observations back into the embryo and try to see how far that matches actually what is happening in the embryo. So it's a very simple experiment what we are doing here, and these simple experiments have been done nearly 100 years ago already on amphibian embryos. And the way we are doing that is we are just taking out ectoderm progenitor cells, primary cells from the embryo. We label them in this case red. We're taking out mesendoderm progenitors, which we label with green. We are dissociating these cells and we are mixing them, and then we are putting them into little valves, culture dish valves here. And then we're watching if they have if they show a self-organizing property in terms of cell segregation. The experiment looks like that. You're putting them in. Red is ectoderm, green is mesendoderm. I hope you can see that actually. The red cells are forming homotypic clusters, which are surrounded by homotypic clusters of mesendoderm cells. There's a very, very clear segregation of these progenitor cells, and ectoderm end up in the middle, and it's being surrounded by mesendoderm. So we can actually use these culture assays now to understand how these cells are segregating, how they are sorting, to eventually understand how that process works in the embryo. I will probably exclusively, for the rest of my talk, speak about these in vitro assays, where we look at cell segregation. cell sorting, as I mentioned that already, is not a very recent invention, and many people have looked at that. And nearly 100 years ago, that has been noted that cells are segregating in embryonic development. And one of the major steps ahead in understanding how cell sorting is done came from work by Steinberg and his colleagues more than 60 years ago now. And what Steinberg was proposing is that tissues do behave like immiscible liquids which have a given surface tension, to some degree similar to, if you want, oil and water. And I believe water has a very high surface tension, oil a lower surface tension. If you put a drop on oil of water, it will spread out. And basically what Steinberg has proposed, and this is really an old hypothesis, he's saying that the surface tension or differences in surface tension between tissues triggers the segregation of cell types and triggers the spatial organization of these different homotypic clusters. And basically the black cells in the simulation, which was done by Glaser and Garnier using their cellular POTS model, basically what they show here in the simulation is that the black cells form a tissue which has a higher surface tension, and the white cells form a tissue which has a lower surface tension. There is some affinity between these two tissues, and what you're ending up with is a black tissue which is surrounded by a white tissue. So knowing and learning about the surface tension of tissues gives you an opportunity to learn something about the segregation behavior of these cells, leading to tissues which have different surface tension. Okay, so the first way to see if surface tension or differences in surface tension between the different germ layer tissues might determine their segregation behavior was actually measuring these properties, tissue surface tension. And what it is, we built a little machine which is very similar to a machine which had been used for many years again. It's a very simple machine which has two metal plates. One is up here, another one is down here. And what you can do is you can take a little piece of germ layer out of the embryo containing perhaps 100 or 200 cells. You put it into a culture dish. You wait till it has really rounded up. It's completely spherical. Then you put it between the two metal plates. And then you're moving the upper plate down and recording the force by which this movement down is resisted. And you're looking at the deformation of this aggregate here, the spherical aggregate. And from the deformation and the force, you calculate then the surface tension. And you can do that for ectoderm tissues, but you can also do it for mesendoderm tissues. And we have done many of these experiments, and what we found is that ectoderm has indeed a higher surface tension than mesendoderm tissue has. And this is consistent with the idea that the tissue which has the higher surface tension goes in the middle and is being surrounded by the tissue which has a lower surface tension. The ectoderm in our in vitro sorting assay goes in the middle and is being surrounded by mesendoderm. So surface tension, the difference in surface tension are consistent with the idea that surface tension determines the segregation behavior of germ layer tissues. Okay, so we thought we are one step further now to understand how these progenitor cells are segregating, till we discovered one feature of the system which was a bit problematic. So when you look at a cluster of progenitor cells, and this is a relatively small cluster, but you can have hundreds of cells, you're putting them into a culture dish, you wait till they round up, and you watch them, and then you do your surface tension measurements. What we observed was that once we took a tissue, in this case it's going to be an ectoderm cluster of cells, and we took it from an embryo which is expressing GFP specifically in epithelial cells, and only in epithelial cells. So we put it in, and we didn't expect very much to happen, but once we left that into culture, what we noted is, and again it's very hard to see for you, there is an epithelial shell surrounding the inner cells very, very shortly after you put these cells into culture. So all the surface cells are organizing an epithelial shell-like structure, and basically you're ending up with a heterotypic cluster of cells which have epithelial cells on the outside, and the inner cells are non-epithelial.
So if you say fast, what's the time
scale? This is a time scale of hours, 3 hours approximately. Which is problematic because the experiment itself takes you 1.5 hours, and you're already in a regime where you have surface cell differentiation. And so if you're starting then to measure surface tension, you don't have a homogeneous population of cells, and it's very hard to predict how these different features, having an epithelial shell and inner cells which are non-epithelial, affect these surface tension measurements. So we thought this is a bit too complicated now, and we have to simplify our assay to learn about the differences in surface tension and how these differences in surface tension are being achieved in the different progenitor cell types. And the simplest aggregate, multicellular aggregate you can think of are two cells. And so we thought that the best way might be we are just looking at homotypic doublets of cells. So we're looking at homotypic ectoderm doublets and comparing them to homotypic mesoderm and endoderm doublets. The advantage of looking at doublets of cells is that you don't have surface cells and inner cells, but there are only two cells which are contacting each other. And what we assumed is, obviously, if you have a doublet of cell, it's very hard to measure their surface tension because it's too small. But what we assumed, if the surface tension of this doublet of cells is increasing, what this doublet will do, it will try to round up, basically. It will take a round shape. And so if we monitor the roundness of this doublet of cells, we learn something about the surface tension of these doublets and possible differences in surface tension between ectoderm, mesoderm, and endoderm, and the underlying cell biology, which leads to rounding up and surface tension of these doublets. And if a cell doublet rounds up, what it does usually, it's increasing its contact area. So basically what we assume is if the surface tension goes up in such a doublet, what these two cells have to do is they have to increase their cell-cell contact area. So if we learn something about cell-cell contact area, rounding up, surface tension, then we will know something about the segregation behavior. Okay, so now here we come, pipettes. If we look at doublets, you have to somehow create these doublets, and the way we are doing that is using micropipettes. These are glass pipettes, as you can see here and here, and they are connected by a tubing to a pump, and the pump creates under pressure in these pipettes. So you have a sucking pressure, and what you do is you take these pipettes, they are on arms, micromanipulators, you move them to the cells in your culture dish, you have to dissociate your primary progenitor cells, and then you pick cells up, and what you can do is you can move them together, as shown in this movie here, and then you can watch them. And it looks pretty chaotic, but you can, if you do it in a systematic way, you can learn something out of it, and you watch how these cells are forming cell-cell contacts. So basically you look at the doublet, and then you can dissociate them again. But you look basically at the doublet, and you watch how this doublet goes into a steady-state configuration where it has a specific contact area which corresponds to the surface tension of this doublet. And we have done that for ectoderm, we have done it for mesoderm as well. The way we are getting these progenitor cells is basically going to embryos which are expressing GFP in different progenitor cell types and isolating these single cells. This is a mesoderm one. You might be already able to see that they're forming a slightly smaller contact, and endoderm cells form usually a very, very small contact. Okay, this is the way we can create doublets, and we can monitor their shape changes. And what we did is to understand if there are differences in the shape of these doublets is we looked at the morphology of a cell doublet. Perhaps this is an example here, and we are not looking at the contact area, but we are looking at the contact angle, because that's independent than the size of the cells which are contacting each other, to avoid that there's any systematic difference in the size between ectoderm, mesoderm, and which there is not, but it's just an easier way of measuring it. So when we're looking at these contacts and the contact angle, what we're finding is that ectoderm doublets over time, over contact time, and this ranges from 1 minute up to 10 minutes, increase their contact area, and it goes into something which looks like steady state. It's not really steady state, but I think they're stabilizing the contact area, contact size at this stage here. And if you compare that to mesoderm and endoderm, they're forming contacts which are significantly small. And this is consistent with our surface tension measurement at these multicellular aggregates, saying that ectoderm is a higher surface tension, and so the doublets forming a larger contact area because they're trying to round up, because they have a higher surface tension, and mesoderm and endoderm cells forming a smaller contact area because they have a lower surface tension. Now, if you look then at these cell-cell contact angles, and basically the question is then why is ectoderm forming a larger cell-cell contact than mesoderm and endoderm? And I'm coming back here to Steinberg again, who had some ideas about how cells are contacting each other and how surface tension is being established. And what Steinberg was proposing in quite sort of well-known, at least in the development biology community, well-known hypothesis, and his differential adhesion hypothesis, what he's saying is that these two cells will increase their contact area mainly because they are more adhesive. and they express more adhesion molecules. That was his simplification. The number of adhesion molecules would determine the size of the contact. So basically what he was proposing is differential adhesion hypothesis, that contact formation and expansion is driven by the binding of adhesion molecules, cadherin adhesion molecules in most cases, which create an adhesion energy which expands the surface of contact. And so we thought to understand if bi-ectoderm doublets form a larger contact than mesoderm and endoderm, perhaps they are more adhesive, and that's the way it can explain why they acquire different surface tension and why they are actually then segregating. So we thought we need to look a bit closer at the differential adhesion hypothesis. To do that, what we did is, and here we teamed up with Frank Jülicher, we looked at cell doublets and thought and asked how is the cell-cell contact between a doublet determined, and it's not only adhesion which determines the doublet shape and the contact area or the contact size, but there is also cortical tension. Cells have an actomizing cortex. The actomizing cortex is contractile, and the contractility of the actomizing cortex is an important factor which determines the size of the contact. And you have to imagine the cortex is not exactly the same. There's a cortex which is formed at the cell. In this case, it would be medium interface or interstitial space interface in the embryo. And there's also a cell-cell cortex which is formed or which is maintained at the cell-cell contact area. And if you look at the contribution of these cortical tensions which are created by these contractile cortices, then they are very different. So if you have cortex tension building up by contraction of actomizing at the cell-cell interface, these cells, once they're making a contact, will try to round up still. So it counteracts the contact expansion, because you get contraction here, and this contraction tries to minimize the contact area. And so cortical tension at the cell-cell interface, gamma CC, would try to reduce the contact size. You have also cortical tension at the cell-medium interface, as I mentioned before, and these cortical tensions don't have to be the same, because there are differences in the cortical organization between these two interfaces. The cortical tension at the cell-medium interface basically acts on the margin of the contact, and you have to imagine these are two contractile shells which are connected here at the contact area. And what cortical tension at the cell-medium interface is trying to expand the contact, because it pulls on the contact till the forces are balanced back here. So you have cortical tension gamma Cm, which is trying to expand. This would be this vector here, which is trying to expand the contact area. You have cortical tension at the cell-cell interface, which is trying to reduce the contact area. And then you have adhesion. And this would be an adhesion tension, which we plot here, omega. And if the doublet is in steady state, if it's not changing its shape anymore, all these forces have to be balanced. And basically by the shape, you can look at the shape, and the shape is determined by the balance of these cortical tensions and the adhesion tension. The question we ask now, are the differences in the shape between these doublets and the difference in the surface tension all determined by omega, which would be the differential adhesion hypothesis saying omega is doing the job, adhesion tension is doing the job, and cortical tension is perhaps not so important. Now, how can we learn something about these different tensions? And the way is another method, which I very shortly introduce here. What we did is here, we want to measure these cortical tensions, and then via the shape of the doublet and the measured cortical tensions, calculate what the adhesion tension would be, and if the adhesion tension is the determining factor determining the shape of these doublets. Now, what we're doing in this experiment here is we're using an atomic force microscope. It has a cantilever. There's a laser which is reflected by the cantilever. We put a plastic bead on the tip of the cantilever. We glue it on the tip of the cantilever. We're putting a cell on a substrate. The substrate doesn't have to be adhesive. This is a single progenitor cell. And basically, it forms a cortex which is at the interface to the medium, because it's a single cell not in contact to another cell. And then we are moving down the cantilever with the plastic bead on its tip. And we are moving onto the surface of the cell, we are deforming the cell, we are watching how the cell is deforming, and we are measuring the force which is created by this deformation. And again, what we can then calculate is the resistance of the contractile actomizing cortex to the movement of the bead trying to deform it. And so what we get from these experiments, we get a cortical tension which is the cortical tension at the cell-medium interface of single progenitor cells. And we can do that for ectoderm progenitor cells, mesoderm, and endoderm progenitor cells. When we did these experiments, what we found is that the cortical tension of ectoderm progenitor cells, single ectoderm progenitor cells at the cell-medium interface is higher than mesoderm and endoderm progenitor cells. So we have a readout for gamma CM, cortical adhesion at the cell-medium interface. That's useful if you go back to the doublets because you have one of the parameters you have already determined. The other parameter is much more difficult, and that is cortical adhesion at the cell-cell interface, because you have no access to the cell-cell interface directly. You cannot deform it because the cells are in contact to each other. One way, and this is a quick and dirty way, and I'll show you a better way then, is just looking at the localization of actin and myosin at these different interfaces. Since we know what the cortical adhesion at the cell-medium interface is, then we can, by looking at the differential distribution of actomyosin, we can look at the ratio of cortical adhesions, assuming that actomyosin would scale this cortical adhesion, which is all a bit shaky. Basically, this is sort of a quick and dirty way. What we are doing here is we are looking at myosin localization. This is a cell doublet, and it looks a bit confusing because you see myosin, this is cortical myosin here, and then you see a bit of cortical myosin here. The reason for that is that these cells are very, very actively blebbing. They're dissociating the plasma membrane from the cortex. That leads to repolarization of the actomyosin cortex. So you have a cortex which is just a bleb which is being retracted here. You have a remaining cell cortex here, another bleb which is not retracted. But basically, these localization of myosin give you an idea, a very rough idea, what the cortex, how much myosin is localized to the cell-medium interface. And you can also look at the cell-cell interface, and you do that obviously not only in one level, but you do it in 3 dimensions. And you see that there's very little cortical myosin left at the cell-cell interface. And if you look at the ratio of these two intensities of the cell-medium and the cell-cell interface, you might learn something about the ratio of cortical adhesions between these interfaces. And since we have measured the cortical adhesion at this interface, we then would learn something about the cortical adhesion at this interface. And what we did these measurements just looking at the intensity of myosin, and what we find is that ectoderm most efficiently gets rid of myosin and actin at the cell-cell interface compared to the cell-medium interface. Mesoderm is less well able to do so, and endoderm not as well. It's similar to mesoderm. So that gives us some idea about the ratio of gamma CM to gamma CC. Another way is a much more accurate way, and that's again, and this is interesting now in the context of a technique lecture here, is using micropipettes again. Now what we are doing here in this experiment is we are creating a triplet of progenitor cells, a homotypic triplet of progenitor cells. These could be all 3 ectoderm or mesoderm or endoderm. And we are holding this triplet with 2 micropipettes, under pressure here, under pressure here. And then what we are doing is we are pulling this triplet apart. And what we want to have is that it breaks either here or here, it doesn't matter. But what you do is you basically dissociate 2 cells from each other. And the reason why we're using triplets is that the cell in the middle is unperturbed by the pipettes, because if you're aspirating these cells, you can see that you get this tongue inside the pipette which deforms the cells, and by deforming the cells, you might change properties like cortical adhesion and adhesion tension of these cells. So we want to look basically at the unperturbed cell, how it changes its shape upon separation of its cell-cell contact to another cell. And the idea is basically that you take this triplet. You're moving this cell away. It breaks perhaps here, this or here, and then you're looking how this cell, at its just exposed cell-cell interface, deforms. And the reason for that is that what we found before by just looking at myosin is that the cell-cell interface has less actomyosin. The cortex is reduced at the cell-cell interface. And if the cortex is reduced, and thereby the cortical adhesion is reduced, you would assume that the pressure cell is uniform. You basically look at the deformation at the cell-cell interface due to its weaker actomyosing cortex at this interface which you are just exposed. And via the deformation, then you learn something about the weakening of the actomyosing cortex at the cell-cell interface, and thereby about the ratio of cortical adhesion at the cell medium to the cell-cell interface. Now the experiment looks like that. You're pulling, and then you see very nicely this deformation at the former cell-cell interface here. So you can do these experiments, and then you can basically calculate from these experiments then the ratio of cortical adhesion at the cell to the cell-medium interface.
Okay, but does the same work if you just don't take no pipettes, but just sell it here on a flat thing, for example?
You mean on a substrate? Or? Mm-hmm. The problem is that if you put cells on a substrate, you have cell-substrate and cell-cell adhesion, which is complicating the thing. I mean, here you want to have selectively look at cell-cell adhesion. And the way is that we want to unbind these cells, separate these cells, and the best way is really keeping them away from the substrate, just holding them as pipettes and putting them apart.
Now, instead of holding the cell in the pipette, you could just stick a cell on a rod.
And then pulling on them?
And then just pulling the other side. Because now it doesn't really, and in this case you don't really care about how much you aspirate the cell.
Yes, I mean, in principle, you do nothing else in an AFM experiment if you would separate cells. I mean, you're gluing the cell via con-A, via lectin to the cantilever. The problem is, I mean, this is, of course, this is disruptive here because you're aspirating cells, but gluing cells with lectin is disrupting the adhesive complexes in the cortical organization to a similar extent, which might be very disturbing for cell-cell contact formation. I think all these, you know, the way you hold the cell is always somehow disturbing the system and that's the way you're doing it with pipettes. And I mean, of course, you could coat the pipette here and then just let the cell stick to it and don't apply pressure. Not sure what is really better in the end.
Yeah, I was just wondering if you see them still the same or if there's some effect that you're...
I mean, importantly, we are not looking at this cell. We're not looking at this cell here, but we're looking only at the cell in the middle. And the cell in the middle is not perturbed by anything. Right? It's not being aspirated. We're basically looking then at this deformation here of this cell. Yeah.
So these experiments and the doublet experiments, is it looks as if the bleb that arises from other parts of the cortex are suppressed as soon as contact is made. So can you...
these cells are blebbing as
having a free edge or actually the contact suppression.
Yes. It's probably right that if cells are forming a cell-cell contact, their blebbing activity is reduced. We have not systematically really analyzed that, but that's an observation we had as well. If you leave them in contact, their blebbing activity goes down. And they're certainly not blebbing directly at the contact because there's no space for blebbing. But there seems to be an effect on cell-cell adhesion on their blebbing activity, which I don't fully understand yet why that is so. Okay, now we use two methods now to calculate what the ratio is of cortical tension at the cell medium interface. Cell medium interface we use with AFM, we can measure that. And now with the triplet assay, we know something about the ratio of the cortical tension at the cell medium and the cell-cell interface. We know the shape of this doublet here, and by the contact angle, and we have two of the parameters experimentally determined, we can then calculate what the adhesion tension is. And Steinberg would propose the adhesion tension is really the driving force and it makes everything different. But if you're looking now at the ratio, for example, of adhesion tension to the cortical tension at the cell medium interface, the contribution of the adhesion tension even appears to be negative here, which is not true, but it's basically very, very small. And that just tells us that the shape of a doublet is, you're talking here about germ layer progenitor cells, and the shape of these germ layer progenitor cell doublets are mainly determined by the ratio of cortical tension at the cell-cell interface to cortical tension at the cell medium interface. This ratio really determines the shape of these progenitor cells, their surface tension, and their segregation behavior consequently. And what the contribution of adhesion tension to this shape of these progenitor cells is really minor. And that's completely different to what Steinberg would have proposed, and what we are proposing here is that the differences in cell-cell contact angle and thereby the surface tension of homotypic doublets between the different progenitor cell types is predominantly determined by the different ratios of cortical tension at the cell medium to the cell-cell interface. That really makes cells different. So basically to put it in more simple words, what makes ectoderm different from mesoderm and endoderm is that ectoderm cells are more better able to disassemble their actomizing cortex as a cell-cell contact area and thereby to expand their contact area in comparison to mesoderm and endoderm progenitor cells. Okay, and adhesion tension only marginally contributes to cell-cell contact formation. The obvious question then is, what is adhesion good for? I mean, obviously, if you don't have any adhesion molecules expressed in these cells, if you knock down the expression of cadherin 1 in these cells, they are not forming any cell-cell contacts. So adhesion certainly has a function in cell-cell contact formation, but it's not by providing adhesion tension. And one function we thought might be the most logical one adhesion is doing is actually binding these cells together at their contact edge. So you have to imagine that cortical tension pulls on the contact, on the cell-cell contact, and these forces which are pulled in opposite directions have to be somehow balanced and connected and coupled at the cell-cell interface. And this coupling function, this mechanical coupling function of these two contractile cortices at the cell-cell contact, this has to be somehow achieved by the adhesion complex, which is linking the two cortices together. Adhesion molecules binding to each other and then coupling to the coating cytoscaling. So what we thought, what adhesion molecules and what adhesion, in this case we are talking about cadherin mediated cell-cell adhesion is doing, it's mainly coupling the contractile cortices together, mainly providing a mechanical scaffold for cortical tension to determine the contact size of these cells. So a mechanical coupling function of adhesion. Okay, here we come back to the pipettes. How can we look at the mechanical, how can we analyze, how can we determine the mechanical coupling function of adhesion, this mechanical function of linking these contractile cortices together? And the way we thought, one way we thought we can do that is actually doing a very similar experiment I showed you before, and that is just exemplified here. And I run this movie, you take a pipette, you hold one progenitor cell here, you hold another progenitor cell here, you're putting them together, then you're waiting for a given contact time that can range between 1 and 10 minutes. And what you do then is you go with this pipette and you try to aspirate it into the pipette, you stepwise increase the pressure in the pipette till you reach a critical pressure where you can pull these cells apart. And if you know the diameter of the pipette, and if you know the pressure between the last step, which was not efficient to separate them, and the next step, which was efficient to separate them, then you basically can calculate the force which is required to separate this doublet. And what we reasoned here is that the force which is required to separate a cell-cell doublet is largely, but certainly not only, but largely determined by the mechanical coupling function of adhesion at the cell-cell contact. Basically, the mechanical function of adhesion gluing these cells together. There are other effects. It depends on the speed you're pulling these cells apart. It depends on the mechanical properties of these cells. But one important parameter is certainly the coupling of these cells at their cell-cell contact. So by determining separation force, we might learn something about the mechanical coupling function of adhesion at the cell-cell contact. Okay, so we have done these experiments for ectoderm doublets, and we have done it for mesoderm doublets and endoderm doublets. And this is basically the contact time ranging from 1 minute, 5 minutes, 10 minutes. And this is the separation force we have determined when we pull these cells apart, which should be somehow a readout or a proxy or an indication for the mechanical coupling function of adhesion. And what you see is if you just look at the separation force, ectoderm cells at varying contact times from 1 up to 10 minutes have a consistently and significantly higher de-adhesion force required to separate these doublets than if you look at homotypic mesoderm doublets and homotypic endoderm doublets. Now, there are a couple of relatively simple reasons why ectoderm cells are more sticky or you need more force to separate them. And one, the simplest explanation would be we know already from the experiments before that ectoderm doublets have a larger cell-cell contact area. And since the area would be larger, you might have more cadherin adhesion molecules, and then it would be trivial to say that it's harder to separate them. So what we did is to exclude that this is actually the explanation by the separation force is higher in ectoderm is we divided the separation force by contact area, getting a stress here, and we're looking then at ectoderm, mesoderm, and endoderm. Still ectoderm is higher than mesoderm and endoderm, indicating that the difference in separation force is not just due to ectoderm having bigger cell-cell contacts. Another simple explanation would be that ectoderm just express more cadherin adhesion molecules, and then you would have more of these mechanical linkers at the cell-cell contact, and that would determine then the separation force. This is not the case, and we knew that already for a while. Looking at the amount of cadherins at the cell-cell contact, and there's one cadherin which is particularly important, this is cadherin 1, E cadherin. We know that ectoderm actually has the smallest amount of adhesion cadherins at their plasma membrane compared to endoderm and mesoderm cells. So certainly ectoderm has not more cadherins at their contact. So the amount of cadherins would not explain, or difference in the amount of cadherins cannot explain the difference in the separation force we have seen there. Okay, so what we needed to do is then we need to understand why these adhesion complexes in ectoderm cells appear to be stronger, more resistant against pulling forces, and therefore we get a higher separation force in ectoderm compared to mesoderm and endoderm. And if you look at cadherin adhesion complexes, and this is probably the most simple representation, I mean, in reality there are approximately 1,000 proteins here and another 1,000 proteins down here. We just look at the absolutely core components. this would be the cadherin adhesion complexes. They are forming trans dimers here, and the cytoplasmic tail of the cadherins are linked to beta-catenin. Beta-catenin binds to alpha-catenin, and alpha-catenin binds to the corticocytoskeleton. The binding of alpha-catenin to actin is disputed, and there might be another intermediate molecules like vinculin and apelin. In the meantime, I think it's again believed that alpha-catenin can actually bind simultaneously to actin and beta-catenin, but the situation is a bit unclear at the moment. But in principle, you have a mechanical link of cadherins via beta-catenin and alpha-catenin to the cytoskeleton. And what we were asking is, if you pull on this complex here, where does it break? To know what determines actually this mechanical coupling function of the adhesion complex. Is it breaking here? Is it determined by the binding affinity of cadherins to each other? Or does it break somewhere down here? Okay, first we looked at the localization of these components just to make sure that everything is in place. And what we find is that cadherins are indeed present. This is an antibody staining. And here we're looking right in the embryo. And what is interesting actually that these cadherins are not just homogeneously distributed over the cell-cell contact, but they're localizing in a ring-like structure at the margin of the cell-cell contact area. So all cadherins are actually at the margin of the cell-cell contact area, where you would imagine the cadherins are actually required, because there they have to couple the contractile cortices pulling on the cell-cell contact area. Okay, now the experiment is, we want to know what is happening if we pull the adhesion complex apart from each other. Can we do that a bit darker? Probably not, right? That's it. A way of getting the lecture hall darker? No. Okay, so you have to believe me, but I tell you now. Okay, what we're expressing here is a GFP coupled cadherin. These are two cells. Cadherins localized at the cell-cell contact. We're pulling them apart. What you find is there's one cell, there's another cell which you can't see. There's a plasma membrane tether connecting these two cells when you pull them apart. And all cadherins, all GFP cadherins at the cell-cell contact flow into this tether. So you're pulling on the complex, on the adhesion complex. What is happening is that the cadherins rupture from the actomizing cytoskeleton and flow into a plasma membrane tether connecting the two separated cells, not fully separated cells. So cadherin apparently is, the link between cadherin and cadherin is fairly strong. You don't break that very easily. If you look at beta-catenin, and again, it's not visible for you, beta-catenin localizes somewhere here. You're pulling on these cells, you're pulling them apart. There's one cell, there's another cell, plasma membrane tether. And in the middle of this tether, this would be the border one cell, in the middle of the tether, you have all beta-catenin pulled in. So basically, somewhere it ruptures downstream of beta-catenin as well. When we look then at alpha-catenin and at actin, and this is all invisible for you, then we don't see alpha-catenin flowing into these tethers and actin flowing into these tethers. Basically, the adhesion complex apparently has its weakest link between beta-catenin and alpha-catenin. To be on the safe side, what we propose here is we say that the coupling of cadherins to the cytoskeleton is weaker than the binding of cadherins to each other. I think that's a very safe conclusion from these experiments. But it's an interesting conclusion because it tells you the mechanical coupling function of cadherins is determined via their ability to bind to the corticocytoskeleton. This is just a summary of these experiments when we are pulling them apart. You're seeing that these are two cells, cadherins, alpha and beta-catenin are localized in the ring-like structure. We're pulling them apart, and then what is happening with the cadherin? Together this beta-catenin flows into these spaces, into these plasma membrane tethers, and it leaves behind the actomizing cortex, in this case, including alpha-catenin. Okay, so we know the weakest link is somewhere from the cytoplasmic tail of cadherins into the cytoskeleton. We wanted to know that if that is something actually we see in the embryo again. We're looking here right in the embryo. We're looking at mesoderm cells which are moving apart from each other, and again, it's very difficult to see for you. But you have to believe me, they are plasma membrane tethers, PPIN cells, which contain cadherins, indicating that when cells are moving apart, separating within the embryo, and they're expressing now a GFP cadherin, cadherins do not dissociate at the contact, but they flow into plasma membrane tether, and somewhere it breaks between the cytoplasmic tails of the cadherins and the corticocytoskeleton. Okay, now the question we ask is, we know that the mechanical coupling function of cadherins is stronger in ectoderm compared to mesoderm in the endoderm. And we know that this coupling function is determined by the anchoring of cations to the corticocytoskeleton. So we assume that ectoderm has a stronger anchoring to the corticocytoskeleton. And we were asking, is this mechanical coupling function or the differences in the mechanical coupling function between the progenitor cells important for their ability to form differences in different sized contacts and differences in surface tension and their segregation behavior. The experiment we are doing here is, again, we're using pipettes. We are putting two progenitor cells together, and then we are watching how they are forming cell-cell contacts. And we are doing that in two different conditions. We are doing it in conditions where we knock down all endogenous cations in these cells and putting back a certain amount of a full-length cation, which we can label or unlabeled full-length cation. We're doing a similar experiment where we're knocking out all endogenous cations in the three progenitor cell types and put back a form of cation which is truncated on its subplasmic tail, which can't bind consequently to the corticocytoskeleton anymore. And then we're asking, is this coupling function of cations important for formation of cell-cell contacts, which I would assume it is, but more importantly, would the differences in cell-cell contact area be still apparent between ectoderm, mesoderm, and endoderm when they have cations which have a similar weak or non-existent coupling to the corticocytoskeleton anymore? This is the experiment. We're putting them together. In this case, we're expressing full-length cation, and we're doing that, and we're comparing here ectoderm and endoderm progenitor cells. Okay, so when we do these experiments, what is happening is usually if we are expressing a full-length, and just look here at the contact angle, the contact size. If you're expressing a full-length cation in ectoderm progenitor cells, then the ectoderm contacts are larger than expressing a full-length cation in endoderm progenitor cells. That's consistent with the endogenous function. If instead we are expressing a truncated version of cations which is not able to bind to the corticocytoskeleton, the contact shrinks considerably in ectoderm progenitor cells, and it has a similar small size than you see in endoderm progenitor cells. And that's actually an important experiment because it tells you that the differences in contact size critically depend on differences in the mechanical coupling function of cations in these different progenitor cell types. I talked about doublets, but eventually you want to see if these the importance of the mechanical coupling function of cations in determining cell-cell contact size is actually sufficient to drive sorting of cells. Now, what we did is an experiment just to test that is taking, in this case, twice ectoderm progenitor cells. And usually, if you mix two ectoderm progenitor cells, there's no segregation because they're the same cell type. Now, what we did is we labeled half of the population red and the other half of the population in green. And in this case, we expressed in both of these populations in the control experiment the same amount of full-length cation 2. And what we expected, what was happening is that there's no segregation of these cell types. They're expressing the same amount of cations, same cell type, nothing is happening. But if we are doing the experiment and just look over here where the red cell is expressing a full-length cation which is able to couple to the corticocytoskeleton, and the green cells are expressing a truncated version of cation which is unable to couple to the corticocytoskeleton, what is happening is that the red cells are sorting in the middle because they're forming larger cell-cell contacts, they're more cohesive, and they're being surrounded by the green cells which form smaller cell-cell contacts, and they're less cohesive. And that tells you really that the coupling, the only difference these cells have is the ability of cations to couple the corticocytoskeleton is sufficient to drive segregation of progenitor cell types. So it's an important function. The adhesion function, which is mechanical coupling, providing a mechanical scaffold, is really critical for progenitor cell sorting. Okay, so just to summarize what I tried to show you here, summarizing a number of different techniques. We have used parallel plate compression apparatus, AFM, pipettes. Basically what we conclude is that E-caterin-mediated adhesion mechanically couples the contractile cortices of contacting progenitor cells. So adhesion is not useless, adhesion is important, but it's not providing adhesion tension, but it's providing a mechanical scaffold for cortical tension to determine the contact size. We show that the coupling strength of E-caterin scales with the cortical contractility. We know that this coupling function is stronger in ectoderm, which has a higher cortical contractility. There's a cortical contractility with different progenitor cell types, and is determined by the cytoskeletal anchoring strength of E-caterin to the cortical cytoskeleton. And we also show that difference in progenitor cell contact formation and cell sorting depend on the cytoskeletal anchoring of E-caterin. And that's it. So I'm pretty much at the end. And just once more, I want to acknowledge Jean-Léon, who has done most of the experiments. Thank you.
So have you tried the nickel-cadmium alpha-cadmium fusion passive?
Yeah, we have tried that as well, and the fusion, it just breaks then at the next step downstream of it. So the fusion doesn't change too much, and it doesn't change this, the contact size between ectoderm on the one side and mesoderm and endoderm on the other side, because it's, I think the fact that the weakest link is between beta alpha-catenin is not very crucial. I think the next weakest link is presumably between alpha-catenin and the actin cytoskeleton, and this is different between the different progenitor cell types equally. I think the critical question really be asking now is what makes this mechanical scaffolding function different between the different progenitor cell types, and I think the easiest way of explaining it would be mechanosensitive function of these adhesion complexes. It has been shown that if you pull on adhesion complexes, they strengthen with the force you're pulling on these complexes, and this comes about by alpha-catenin unfolding, revealing vinculin binding sites, or vinculin unfolding, revealing actin binding sites. And since we know that the total cortical tension is higher in ectoderm progenitor cells, so that pulling with a stronger force on the adhesion complex, this might just strengthen the adhesion complex, strengthen their mechanical coupling to the cortical cytoskeleton. This might be the reason why we are seeing a stronger coupling to the cytoskeleton.
There's one little thing I didn't understand in one of the last slides where you showed the differential capability of the different coherent molecules to induce cell sorting or not.
Yeah.
The ectoderm and this one. So I understand for the ectoderm because you showed in an earlier slide that the full-length coherent and the truncated coherent, they have this differential adhesion, right? That it has a large effect. What I don't understand is on the endoderm.
It's actually true. So if you just look at the contact area, endoderm doesn't reduce the contact area much if you're expressing a truncated versus a full-length coherent. Still, these cells are segregating, but the segregation is by far not as pronounced as you have it in an ectoderm case. So what we believe is there's still a change in the contact area, which is below our detection limit. I mean, if you look at these, the problem is always if you determine the contact area, you have these very, very bloody cells which are changing constantly, and what you get is a mean of a contact area which has a lot of noise, basically. And the only explanation we have here for endoderm is that there is a reduction in the contact area which is below our detection limits. But it's a very good observation. It's absolutely right.
So, could the differences be due to just levels of either alpha or beta-catenin?
So we quantified all that. Of course, I mean that would be a simple explanation. Either caterans or alpha or beta-catenin, they are all expressed in very similar levels, or even in an opposing, in a way which would be not consistent with a stronger mechanical coupling function. I think alpha and beta-catenin are actually expressed in very similar levels in all three progenitor cell types. And E-caterin, which is a critical one, is higher expressed in the mesoderm and endoderm, where you would expect they should be lower expressed.
But if the difference is you're invoking a kind of some kind of feedback mechanism, if I understood correctly at the end. But doesn't there have to be something different going into those amplifiers to get an output that's different?
So the mechanical sensation would not lead to an accumulation of alpha-catenin or beta-catenin. The mechanical sensation would lead to a conformational change of alpha-catenin revealing vinculin binding sites. And then it would be interesting to see if we find more vinculin, for example, at these junctions, because that should accumulate then. Alpha and beta-catenin, not vinculin, we have not checked yet because we don't have a good construct. But that's something you would assume that the binding accumulation of vinculin is increased, and then you should get a better binding.
So you're invoking the different properties of that alpha or beta-catenin.
So it has been shown. This is not our work. It has been shown already by a Japanese group 2-3 years ago that if you pull on alpha-catenin, it unfolds, reveals a vinculin binding site, and that strengthens the adhesion complex. Or adhesion, I think, very general. And similar studies have been done on vinculin, I believe, which when you pull on it, it unfolds and reveals a binding site for acting.
So are you hypothesizing that the critical force for unfolding is different in the different cell types or something like that?
Not necessarily. I mean, you would assume they're expressing the same molecules, But the cortical tension, what is different between the different gender cell types is the cortical tension. As I showed you very much at the beginning in the AFM experiment, the cortical cytoskeleton is more contractile in ectoderm compared to mesoderm and endoderm. So you have a higher force pulling on the adhesion complex in ectoderm cells compared to mesoderm and endoderm. That would lead in ectoderm to a more efficient unfolding of alpha-catenin revealing vinculin binding sites and thereby strengthening. And that would be one possibility. It's all speculation. But what you would expect, and that's very right, you should see eventually more vinculin if vinculin is the critical one, which we have not checked.
So if the difference, or the specialty of the ectodermal cells is that they make this patch free of myosin or less tension, what sets the size of that patch then?
So why are ectoderm actually able to disassemble more efficiently the ectomyzing cortex that cells are contact?
Also, if they do it, like over which area do they decide to do it? Or there must be some self-organizing.
Yeah, this again is something which has been studied by Jim Nelson and other people for quite a while. Is that okay? So if you look at caterans have different functions. There are basically three functions you could imagine caterans have. One function is providing adhesion tension. We show in our system is not very important. The other function would be providing a mechanical coupling function, and the third function is a signaling function of caterans. And the signaling function of caterans, and that has been studied by Nelson and other people, is acting onto the composition of the cortical cytoskeleton, and has been shown that signaling from caterans lead to the localized disassembly of the actomizing cytoskeleton at the cell-cell contact area. And this goes via small GTPases, Rho-REC, and they are being sequentially activated, but eventually it leads to a disassembly of actomizing. What we propose, the best explanation of the easy to speculate, but what is happening actually is that what caterans is doing is they are reducing actomycin to a similar degree, to a similar low degree at the cell-cell contact area in all three progenitor cell types. But what is really different is that there's a more contractile actomycin cortex to start with in ectoderm compared to mesoderm and endoderm. Therefore, the difference becomes bigger. I think the signaling function is not the limiting step. I think they are just reducing it to a similar, very low degree at the cell-cell contact area, but you leave more contractile cortex at the cell-medium interface in ectoderm because it is more contractile from the beginning on. That, I think, would determine the difference.
So you're pushing it all the way back to the actomycin.
Yes.
Explain the differences. So you started at cateran and worked backwards, and now you're saying.
So we have two steps. So one step would be, what we say is that it's all about cortical tension in the end. It's all about the difference of cortical tension at cell medium to the cell interface, which determines. And what caterans are doing is they're basically mechanically coupling the contractile cortices together, and they're providing if you want, so a cortical linker, which links these two contractile cortices together. But in the end, what determines the size of the contact and the sorting behavior of cells is the ratio of cortical tension at the cell medium to the cell interface. That's the idea. And the signaling function of cateran, I think it's efficient enough to downregulate to similar degrees in all three progenitor cell types, but you have a more contractile cortex in the ectoderm to start with.
So is the cell volume different between these two types or three types?
No.
Okay, any more questions? Then, thanks a lot. So let's start.